Hi everyone, Dr. Hinkey here with a lecture video on Chapter 9 on the cell cycle and mitosis. This is the last chapter in this unit, and then we'll have an exam coming up. And then we'll go into a little bit, we'll start looking at how cells actually function, what our DNA does, and that process, starting with meiosis. It's a similar process to mitosis. But I really recommend you focus on Chapter 9. So this is a good thing that they are separated out on our exams, with Chapter 9 and mitosis coming on the next exam, and then meiosis afterward. They are similar processes, so it's really, even the vocabulary is similar, it's really easy to confuse them. So make sure you're really solid on mitosis before you venture into meiosis, and that will really help you to keep them straight. So Chapter 9, the cell cycle and mitosis. We'll look at the cell cycle. We'll zoom in on the eukaryotic chromosome, and then the specific steps of mitosis and cytokinesis. We'll take a look at what bacteria, how prokaryotes divide. And then we'll look a little bit at the cell cycle and cancer. So when we're talking about mitosis, a lot of people think cell replication. It's the entire cell cycle results in, at the end of the cycle, in one cell becoming into two, dividing or replicating itself. So in this process, DNA makes a complete copy of itself and then divides it between two daughter cells. The result of mitosis are two identical cells. Each one is identical to the parent cell. So they are identical to each other. This is cloning. So why might cells need to replicate? Why would I need an exact copy of a cell? Well, if you think about this, we all start as one single cell. We get half of our chromosomes from dad and the other half from mom. Those are joined at fertilization when the sperm and the egg fuse, giving us all of our DNA. And then we're a single cell. As we learned in Chapter 4 on cells, there is a limit to how big a cell can get based on the surface to volume ratio. That's just, if we get too big, then our volume is so great that molecules getting in and out have farther to travel to get from the surface to somewhere in the center of the cell where they're needed. And if energy efficiency-wise, we stop getting enough energy out of our food molecules to move those nutrients to where they're needed, or we can't move the waste out fast enough because it's too far. So we have a limit to how big a single cell can get. So we are not single-celled organisms. On average, a human body is made up of about 35 trillion cells. How did we go from that single cell to a 35 trillion celled individual? Mitosis. Right, so cell replication, that single cell made an exact copy of itself. We became two cells. Then we became four. Then those four copied and underwent mitosis. We became 8, 16, and on and on and on. At some point, we reach adulthood and maturity and are full size, but we don't stop undergoing mitosis because cells still have to be replaced. They get old, they get worn out. Our skin cells are constantly sloughing off, so we constantly need to replace those. When we get cut or injured, we replace those damaged cells, that damaged tissue, through the process of mitosis. So mitosis is used for growth, to repair and replace damaged cells, and it can also, for single-celled organisms, be used for reproduction. So many single-celled protists will undergo mitosis. They'll make an exact clone of themselves, and that's how their population increases in size. So that DNA making a complete copy of itself, here our first bullet point. Let's take a look at our DNA. In eukaryotes, our DNA is arranged as chromosomes. These are very long threads of DNA. So in between, when our cells are just doing their job, they're just these very big, long, stretched out threads. And they're organized, our chromosomes we take that DNA and we organize it around proteins called histones. The DNA wrapped around those proteins to organize a little bit is called chromatin. So chromatin is the chromosomes, the DNA plus the proteins that help to organize it. And each species has a characteristic chromosome number. And we generically refer to that as 2N. And we say 2N because we got 1/2, or N, number of chromosomes from mom, and N, number of chromosomes from dad. N plus N, 2N. And we use that generically because that applies to any sexually reproducing organism. So we don't have to worry about a specific number. If we used 46, we would really be referring to humans. There are a couple other species that have 46 chromosomes, but the size and the length and the genes carried on those differs from humans, so they are distinct. If we used 20, we'd only be talking about corn or goldfish. 94. So 2N and N, those numbers. 2N is also called the diploid number, diploid, and haploid. I always associate that with half the number, haploid, half the number, N plus N. So this is the total number of chromosomes that we have, 2N, in any adult or any organism is 2N. Pause so I can let the dog in. Hi, dear. Oh, that's why he was barking. I'm in the middle of recording and he started barking up. Okay. All right, so when we talk about our chromosomes, if we zoom right in, our DNA, as we learned when we looked at the biomolecules, is a double helix made of repeating units called nucleotides. So here's our DNA, and then we wrap it around these histones, these proteins that help to organize it. And then we kind of loop that up and curl it all up. This image, what we usually associate with chromosomes, only occurs for a very short time in our cells, and we're going to look at when that is. Normally, we're kind of all spread out like this, just a mass. And what I like to think of it as is yarn. So if I just took all the yarn, if I had all kinds of yarn, I had 46 strings of yarn, I could just throw them all over the room. And if I tried to divide them, I would have a snarled, tangled mess. So that's not how we buy or how we package yarn. So we package it into skeins. So a skein would look like, just like 1/2 of these, this nice wrapped and wound and organized bundle. And if I had 46 skeins of yarn in my living room, I could very easily divide them up. So this packaging here in this little X, this occurs at a specific stage. This is after we've duplicated our chromosomes to get ready to divide. And then we're going to separate these two out. So we'll look more closely, but This is sort of the shape, this X shape is the shape we associate with chromosomes. That's after duplication. During most of our life, other than when our cell is dividing, it's just this big mess of thread that's all tangled up in the nucleus. So our chromosomes, think about those 46 chromosomes in humans, or our 2N diploid number. We have 23 distinct chromosomes, and we number them 1, 2, 3, 4, 5, up to 23. 1 through 22 we call the autosomes, or somatic chromosomes. These are the ones that dictate pretty much everything about our body. And we get one number 1 from mom, and we get one number 1 from dad. Those 2 number 1 chromosomes are of equal length. They carry similar genes, and they have their center point is at the same point on both of these. Number 2 chromosome, we get one from mom, one from dad. Code for the same genes. They have a connecting center point at the same location. We're going to look more at all those specific details coming up. But for each of those 22 autosomes, they are identical in length and they carry the same genes. We have 2 of each. So we have 23 pairs of chromosomes, 23 from mom, 23 from dad, that are paired up. They both carry identical information or similar information for our traits. And then the last pair are XY chromosomes, referred to as the sex chromosomes, because they determine whether you're male or female. And they are our only chromosome pair that differ in length. So this is one pair where they're not necessarily the same length. So there's our number, our diploid number, or 2N, because we get N from mom and 1 N from dad. So this is going to be really important. So keeping track of the chromosome number, it's important to get this terminology right about diploid or haploid, and that we have these similar chromosomes. Half of our chromosomes are similar to the other half because we got one of each, one from mom, one from dad. We call those homologous chromosomes. So number 1 from mom is homologous or similar to number 1 from dad. Number 2 from mom is homologous to number 2 from dad. They're similar. All right, so when we look at our cell cycle, the cell cycle, this is kind of like a clock. We go in a circle, we come full circle, similar to our cycles that we went through in the Calvin cycle and the citric acid cycle. We're going to come full circle and then start all over again. So the cell cycle is an orderly set of stages that take place between the time a eukaryotic cell divides and the time the resulting daughter cells divide. So I start with one cell. It goes through one cycle of its life, finishing with division, resulting in 2 daughter cells. Now each of these 2 daughter cells will go through those same stages, and in the end, each one will be 2, so it'll be 4 cells. So that is the cell cycle. In my cell cycle, I start here when I present this, and I have the G1 phase, or the growth 1 phase. Some old textbooks refer to this as the gap 1 phase, and it was given that nomenclature of gap because under the microscope with the technology they had at the time when the cycle was being worked out, couldn't see anything going on. So it looked, for all intents and purposes, like the cell was doing nothing. So it was a gap. Now we refer to it as the growth phase because the cell is really, really active. This is where the cell does its job. So this phase is critical. Here, every cell has a function. Muscle cells contract and relax so that we can move. Nerve cells receive stimulus and pass a signal to our brain so that we can respond, send the response back. So they have jobs to do, and this is where they do their job. So really, this is misleading. It looks like these phases are spent in equal time, but really most of the time is probably in G1. And then after the cell's been doing this for a while, it gets a signal that says, we should probably divide now. It's time to grow. And they will enter the S phase. S is for synthesis, because here the cell is going to synthesize an exact copy of its DNA. So DNA replication happens here. Right? And we think we're going to end up with two daughter cells that are identical in appearance, in function, in everything they do. They're identical to the cell we started with. So to do that, I need to make copies of everything. So here I am going to synthesize my DNA. I'm going to make an exact copy. This is where I get this structure. So really, in the G1 phase, this would be a single side of this X. Synthesis gives me this second exact same matching copy. Now I have an exact copy. I go into my growth 2 phase. My growth 2 phase, I make my final preparations for division. Here, a lot of my organelles are going to double or increase in size so that I can divide them between two daughter cells. My mitochondria will double so that I can divide them between two daughter cells. And I'm going to make proteins that I need in order to divide. And some of the proteins I need are enzymes. Some of these are to organize and help divide my DNA into two. So all of this portion here, the cell is not really dividing. It's doing its job, and then it gets ready to divide. So G1, S, and G2 comprise interphase. Interphase is what happens in between cell divisions when the cell is doing its job and then getting ready to divide. Then we enter into the mitotic phase, or mitosis. This is where the cell actually goes through the steps of dividing, and we'll look closer at each of these steps in mitosis: prophase, metaphase, anaphase, telophase. And then this final step, it's considered part of mitosis because this is the part cytokinesis. Cyto, that prefix means cell, kinesis is motion. Here's where I actually divide my cell components into. So the cytoplasm and all the organelles divide. My plasma membrane separates into two cells. The actual mitosis phase from prophase to telophase, this is technically mitosis, and it is defined as nuclear division. So these steps here all take place in a single cell, and at the end I end up with two nuclei in one cell. Cytokinesis divides the rest of the cell around those two nuclei. So those are my steps in my cell cycle. We're going to go through those more slowly, and we'll look at these checkpoints. These checkpoints are just ways our cells protect our body and protect ourselves. They double check to make sure that everything that was supposed to have happened in each phase happens. So after cell division, I will stop and do a check. Did the DNA divide correctly? Was everything divvied up? Do my two daughter cells have the DNA that they need? Was it divided? Was it split? Are there any errors? If everything is good, we continue. If not, I will destroy this cell because something is faulty in it. At G2, I have a second checkpoint before I go on to actually divide my nucleus. We're going to double check if in the synthesis phase, all the DNA was copied correctly. Did it all make an exact copy? Do I have two complete sets of DNA with no errors so that I can divide it evenly between two daughter cells? If not, this cell will destroy itself so that those errors are not passed on. And then in mitosis at metaphase, in the middle of this process, I'm going to stop for another checkpoint to make sure that my chromosomes are lined up and connected correctly to the spindle, to the portion of the cell that's going to divide everything. Make sure we're lined up, everybody is connected, and if not, the cell will destroy itself so that no errors are passed on. So I have some control to make sure that everything happens in the orderly fashion that it's supposed to in the cell cycle. So my two major stages are interphase and mitosis. Interphase is everything where we're not dividing the nucleus or the cytoplasm. So G1, S, G2. And then mitosis, and I'll put cytokinesis as a subset of mitosis, but mitosis is nuclear division. I divide my nucleus. So if we look at the timing, most of the cell cycle is spent in interphase because this is where my cell is actually doing its job. So in G1, I'm going to recover from that previous division. If I divide my cell into two, it has reduced in size. So I'm going to start to accumulate raw materials. How do I do this? Eating, nutrition. I pull in raw materials from the environment and I accumulate them in my cytoplasm. I bring in water so my cell can grow, and I start making phospholipids so my plasma membrane can grow. I start to double my number of organelles. My cell gets bigger, and while it's doing this, it's doing whatever its job is. Then I enter my S phase for DNA replication or DNA synthesis. I enter with one chromosome for each of my 2N. So for humans, I enter with 46 chromosomes. And I make an identical copy. When I make that identical copy, we call each side a chromatid. So I have two identical chromatids. I call them sister chromatids. They are identical to each other. They carry the exact same information, and they are stuck together. They're held together at the centromere. It's sort of a central point where those two stick together. If I entered the S phase with 2N chromosomes, and I make a copy of each one, I still have the same amount of information. So I still say I have 2N chromosomes, but now they are duplicated. I have 2N duplicated chromosomes. I have the diploid number of duplicated chromosomes or sister chromatids. So I like to kind of explain this as a Xerox machine. If I had one piece of paper to give you for a homework assignment, and you were to complete that one piece of paper for your homework assignment, and I took it to the Xerox machine and I made a second copy of it, and I gave you both of those, how many pages of homework would you have? You'd still have one, but now you would have two copies of that same assignment so that you could go and give it to someone else in the class who wasn't there maybe, so that you can share that same assignment with somebody else. So the sister chromatids, they're a Xerox copy. They don't add any information. They can't be used to do anything the cell can't already do. So it's just a copy, and it's just going along for the ride so that later on it can be shared between two daughter cells. So I now have the same number of chromosomes that I came in with, but now each one has a copy stuck onto it, a Xerox copy stuck onto it. Once I've copied those chromosomes, I will go into the G2 phase. And here I will synthesize the proteins. I'll get ready to divide. So I'll make sure I have enough materials. I'll accumulate and make more materials if I need to make sure I can divide everything evenly between my daughter cells. The mitotic stage, mitosis itself, is nuclear division. Those sister chromatids, or daughter chromosomes, sister chromatids are distributed. That should be sister chromosomes. Sorry. Sister chromosomes. Oh, can't stand it. Let me change it. Sister chromosomes are divided between the two daughter cells. So the sister chromatids divide between two daughter cells in mitosis, or between two daughter nuclei. And then I divide the cells around those nuclei. So the cytoplasm and the organelles divide. And I end up with two genetically identical daughters. So before mitosis begins, the very end of G2, the chromatin, which is those two sister chromatids attached at the centromere, starts to coil into distinctly visible chromosomes. So that's where it begins. It's going to take place right up until I enter the first stage of mitosis, so it won't be completed until I'm in prophase, but it's going to condense. And that means it's just going to wrap itself around all those proteins to get organized into these two nice identical. So if here's one skein of yarn, here's the other one. They're attached at the centromere. They're identical. Right? So this is a Xerox copy. I've added no new information to my cell. I've just made a copy of the information I have so I can take that Xerox and hand it off to a daughter cell. So mitosis has 4 stages. Our textbook is going to divide prophase into early, middle prophase, and then late prophase, prometaphase, early metaphase. So talking about this in 4 stages, it seems very stepwise, but it's continuous. So where's the exact hard line from prophase to metaphase? It's kind of blurry because this is a continuous process. This is a very active process that occurs in a continuous progression. So it's not, okay, we get to here and we stop, and now we're in this phase. We get to here and stop. So this is continuous. So the main phases are prophase, metaphase, anaphase, telophase. Sort of a mnemonic way to remember the order is I passed my anatomy test. I would be interphase that comes before this, and then passed my anatomy test. Prophase, metaphase, anaphase, telophase. All right, so as we're getting ready to do this, we have to get some parts in place, or as we enter prophase, we get parts in place to do this. And if you remember our centrosome, the centrioles were those 2 barrel-shaped bodies that are perpendicular to each other. We said we're the mitotic spindle organizing center. They're involved in organizing and building parts of the cytoskeleton. Those are just outside the nucleus. It's our microtubule. I'm going to use my microtubules, those parts of the cytoskeleton, to make the spindle. These are going to be the parts of the cell that are going to move and separate. They're going to latch on to those chromosomes and pull them apart. So lots and lots of fibers composed of microtubules. Those centrioles are only found in animals, not plants. Those were the barrels oriented at right angles to each other. And when my chromosomes doubled, my centrosome was also replicated. So the whole centrosome, both of those barrel-shaped centrioles. So they continue to be in that perpendicular to each other shape, but now I have 2 of them. So what happens at the beginning of prophase is the chromatin finishes condensing. It is now in that visible X form. So I can see the chromosomes with a microscope at this stage. The chromatids are attached at the centromere. The nucleolus, that dense portion inside the nucleus where I was building the components for my ribosomes, the ribosomal RNA, and the proteins that are going to go on to become ribosomes, that just kind of dissolves, that floats away into the cytoplasm because my nuclear envelope breaks up. So I no longer have this membrane-bound nucleus. My DNA is now here in the cytoplasm, as are the parts of the nucleolus. So then I'm in the hard middle of prophase. So this is early prophase. We are still in prophase when we've completed condensing. The nuclear envelope is completely disappeared. The pieces of it, those phospholipids are out floating around in the cytoplasm, and the spindle is forming. The spindle, this is formed by those microtubules, and I like to think of it as a whole bunch of fishing lines that are all going to get connected to either the chromosomes or to other parts of the cytoskeleton right underneath the membrane to anchor them in place, because then they're going to pull those chromosomes apart. So it's like a fishing line reeling in a fish. We see something, we call it the aster. Right? And what that is where those centrioles, our centrosome, the barrel-shaped perpendicular forms, they start to grow these spindles. And it looks like a little starburst or an aster. That's Latin for stars. And those centrosomes move away from each other toward opposite poles of the cell. At the end of prophase, as I'm getting ready to go into metaphase, so pro-metaphase, early metaphase, late prophase, the centromere of each chromosome, so that's the point where the two sister chromatids are joined, develop two kinetochores. And we'll take a look at that in a second. So the kinetochores, if we're thinking of our spindles as being a fishing line, and they're going to grab onto those chromosomes to pull them apart, the kinetochore would be where the hook of that fishing line grabs on to each of the sister chromatids. The kinetochore is a specialized protein complex. There's one on each of the sister chromatids, and that's going to let us pull them apart. So here are our sister chromatids. They are connected or joined together at the centromere. Each one gets a kinetochore. So let's look at those phases we just talked about and what that looks like. So early prophase, we see, oh, look, my chromosomes are becoming visible. They're condensing. This is a really bad picture because they should already be in that X shape. The nuclear envelope has disappeared, and here are my centrioles. So the centrosome was copied in G2, and now it's building this microtubule. These spindles are coming out, and these are moving to opposite poles, so they are separating out. These spindles, these microtubules, some are going to attach to parts of the cytoskeleton right underneath the plasma membrane to anchor it in place, and others are going to attach to the chromosomes. So this is early prophase. Chromosomes condense, nuclear envelope dissolves, and the centrosomes start to move to opposite poles. In the center, in the middle of prophase, my chromosomes are very visible now, and we can see there is no nuclear envelope around that, but we see these fibers, these spindles, reaching out and starting to find those sister chromatids. So we're still moving to opposite poles, but we are now going to start latching on to these, to the chromosomes. And here we see each kinetochore is attached to a spindle from the spindle fiber. And for each sister, each duplicate pair, one kinetochore is attached to one of the centrioles, and one is attached to a spindle from the other at opposite ends or opposite poles of my cell. So we can kind of see what we're being lined up to do here. This is what that looks like under a microscope. As you can see, that looks like a little starburst there where those spindle fibers are. The next phase, metaphase, remember this as middle. So the metaphase, the sister chromatids are aligned in the center of the cell. Those kinetochore fibers, the spindle fibers grabbing onto the kinetochore, are going to push and pull until they've lined up the sister chromatids so that those centromeres are all in a row on the metaphase plate. So that's the equatorial plane or the center of my cell. And remember, this is in the pictures, we're looking at it in 2 dimensions, but this is 3 dimensions. It'd be like if you cut a tennis ball in half and opened it. It'd be all along that surface. The metaphase plate, this is where the mother cell is going to be divided. So I could draw a line right through that metaphase plate, right through all of the centromeres for each of those daughter cells. And that would tell me, oh, this is the direction we're going to divide. So metaphase, my sister chromatids are lined up in the center of the cell. Anaphase, I remember this is A is apart. The sister chromatids are pulled apart to opposite poles of the cell. So the centromere dissolves. That releases the sister chromatids. They are now separate. So now we call them daughter chromosomes, not chromatids. They're no longer attached to each other, and they're being pulled apart from each other. And the final phase is telophase. This prefix television is seeing from afar. Telephone is hearing from afar, or sound from afar. So this is far. In this phase, my chromosomes are at the farthest poles of that cell. They're completely separated from each other. The spindle now breaks down, it disappears, and I have 2 clusters of daughter chromosomes at opposite ends. 46 chromosomes on one side, 46 on the other. And a nuclear envelope begins to reform around those clusters, so giving me the start of my 2 daughter nuclei. The chromosomes start to uncondense, uncoil, and become chromatin again, and the nucleolus starts to reappear. So we're going to start getting those components for my ribosomes back together, because we need those ribosomes to build the proteins so the cells can function. So here's what we look like here. Metaphase. My sister chromatids are all lined up, and I can draw a line right through this centromere, through this one, through this one. That is my dividing line. That's where my cell is going to divide. These spindles are going to start to pull this chromatid this way, this one this way. The one on the left goes left, the one on the right goes right. So we pull them apart. That's anaphase. So here the fishing pole has grabbed on, the hook is here in the kinetochore, and now we're going to reel in the chromosomes to opposite sides of the cell. And then telophase, we're at opposite ends. We're going to start to reform this nuclear envelope. Oh, look, I have 2 centrioles. So when my cell divides, each one will have a centriole. My cytoplasm begins to divide. In telophase, the other thing I get is the beginning of cytokinesis. This is called a cleavage furrow. Oop. My cell gets some cleavage here as it starts to pinch in. If you think about a belt being around the center of the cell and just squeezing tighter and tighter until these membranes touch, and then I have completely divided. So my cytoskeleton is the belt that's going to pull these together. So cytokinesis, the actual division of the cytoplasm, divides the cytoplasm from the original parent cell or mother cell equally around each of those daughter nuclei. And then it encloses, results in each new cell being enclosed in its own membrane. In animals, I get that cleavage furrow as the membrane starts to pinch in to separate out the 2 nuclei. And I get a contractile ring of actin filaments, that cytoskeleton component of actin. It's like pulling a drawstring, and it's going to eventually pinch those 2 daughter cells apart. And at the same time, those actin fibers start to elongate and spread out the cell to help that happen. In plants, we have a little problem with that because we have a cell wall. Our cellulose cell wall is rigid, and so the membrane can't furrow because we need to have this stretching out to pull this apart in this process. So instead, what plants do is they form a cell plate. So the cell will make lots and lots of little membrane-bound vesicles. Remember, that's how we transport stuff around our cell, and what each of those vesicles carries is cellulose. Each of those vesicles is made out of phospholipids, right? They're membranes. So if you can think of where this is going, cellulose inside, surrounded by phospholipid bilayer. These membrane-bound vesicles are all going to line up, and eventually they're going to fuse together. The membrane portion will become the plasma membrane, and the contents of that vesicle become the middle lamella. This is going to be cell wall. It's going to start to, this is a precursor to cellulose, and it's going to secrete that cellulose cell wall. So here's what that looks like. All of these separate vesicles, and once they merge, I have membrane on this side, I have membrane on this side, and all that middle stuff, the lamella starts to produce that cell wall, and that divides my two nuclei into two daughter cells. All right, so we talked very briefly about the control of the cell cycle. So what happens, lots and lots of moving parts in this process, lots of opportunity for things to go wrong. So what would happen if something went wrong in this process? Well, we have a way to control it. So control of the cell cycle, a signal, we have signaling molecules that influence the activity of a cell. The cell cycle is controlled by both internal and external signals. Some external signals are growth factors. They signal proteins on the plasma membrane and say, hey, it's time to divide. Or, hey, our density is enough right now. We have more than enough cells. Stop dividing. We can also have internal signals, a family of proteins called cyclins that are produced by the cell, and they increase and decrease throughout the cell cycle. And that's sort of what gives the cell the message that, hey, it's time to move on from the G1 phase. It's time to divide, so we'd better go through the S phase in G2. So we have to have the right abundance or concentration of these cyclins to tell us when it's time to proceed. If we don't have them, the cell doesn't know that it's time to move on, and the cycle will stop at one of those checkpoints, the G1 checkpoint, the M checkpoint, or the G2 checkpoint. Some cells are always stuck. So for example, nerve cells, we don't go through mitosis with our nerve cells. Once we have our nerve cells, they can grow, they can add new dendrites, new projections, new synapses, new connections from the same cell, but we don't get new nerve cells. That's why nerve damage, brain damage, any sort of nervous system damage is so critical and so difficult to heal because we don't undergo mitosis. Nerve cells enter the G1 phase, and then we don't get a cyclin message saying continue on. We get the message saying, hey, you're good right here. Just keep doing what you do. Do your job. Cells that enter that G1 phase and never make a copy of themselves, never undergo mitosis, are said to be in G0. They're never going to move up beyond this growth phase. They're not going to make a copy. They're not going to go through mitosis. If they are damaged, they will not be replaced. So these checkpoints are looking for these signals. And those checkpoints are also looking to make sure that everything in the previous step of the process happened as it was supposed to. We refer to apoptosis as programmed cell death. Apoptosis and mitosis are opposing forces. One increases the number of cells, the other decreases them by killing them. Together, they maintain homeostasis. They keep the number of cells at an appropriate level. If I have too many, some will die. If I have errors, that would result in something being not in balance, not working properly. I will get rid of those cells. So here are some normal cells. If something went wrong in those cells during the cell cycle, we'd get a chemical signal saying, hey, time to undergo apoptosis. In an apoptosis, the cell self-destructs. And it does that with the help of those lysosomes. So remember, those were the little vesicles that were produced by the endoplasmic reticulum that contained enzyme that would degrade organic molecules. Cell is all organic molecules, so it will produce lots and lots of those lysosomes and then open them up and release that enzyme out into the cell, and that cell will be destroyed. All right, so that's eukaryotic cell division and mitosis, the cell cycle. Prokaryotes, bacteria, go through similar process, but they don't have mitosis. So think about that for a minute. Why wouldn't a prokaryote undergo mitosis? What was the definition of mitosis? Nuclear division. Right. Prokaryotes don't have nuclei. So they go through all those steps, similar process, but we're not going to divide the nuclei. We're just going to copy our chromosome and elongate and divide. So remember, our prokaryotic chromosome was a single ring of DNA just floating in the cytoplasm. Wherever it's floating, we call the nucleoid region. Just like in eukaryotes in the S phase, we synthesize an exact copy of all of our DNA. We are going to replicate that ring of DNA into 2 rings prior to division in our bacteria. Those 2 rings are going to attach to the plasma membrane, a protein in the membrane. They're going to latch on, and they're going to then divide through a process called binary fission. So fission is splitting binary into 2. We're going to split those 2 replicated chromosomes into 2 daughter cells. And the 2 daughter cells would be exact copies of the cell we started with. They will be clones. This is asexual reproduction. And the process is, here's my original cell. This is its full size. There's its DNA. It replicates that DNA. Each of those are connected to the bacterial cell wall, the plasma membrane rather, and the bacteria starts producing, well, here, when it replicates, it continues to build new plasma membrane and new cell wall. So it's elongating. It's getting bigger by bringing in nutrients, bringing in raw materials, and building more and more membrane and more cell wall to get bigger. And that cell wall, it'll start to build it inward. The plasma membrane will start to pinch inward. We'll build that wall and continue to construct that peptidoglycan cell wall and divide the cell until that cell wall meets and divide into 2 daughter cells, each identical to the original parent cell. So it's binary fission. So whether we're looking at binary fission or the cell cycle and mitosis, our functions, so prokaryotes, we have cell division by binary fission. In eukaryotes, we have mitosis and cytokinesis. In prokaryotes, the function of binary fission, of cell division, is asexual reproduction. Bacteria don't get bigger in size. When we say an increase, we are talking about increase in numbers, increase in population. So if bacteria are getting bigger, it's their population that's increasing. So this is asexual reproduction. Cell division in eukaryotes, well, if in some single-cell organisms, so in my protists and in single-celled fungus, I also use mitosis as a form of asexual reproduction. In multicellular plants and animals, and in protists that reproduce sexually. Nope. Only the algae, only the multicellular, and the protists that are multicellular, the algae. They're going to use mitosis and cytokinesis to grow, to increase in size by increasing the number of cells, repair and replace damaged cells, and to develop. So mitosis is cellular replication in most eukaryotes. It is not sexual reproduction. So I'm dividing my cells, making exact copies of them. It is not sexual reproduction, right? Only in single-celled organisms is mitosis used as asexual reproduction. Typically, its primary purpose, when we're talking about humans, about animals, it's growth, repair, replacement of damaged cells. All right, so speaking of damaged cells, what does the cell cycle have to do with cancer? So an abnormal growth of cells, meaning lots and lots and lots of cells, it's called neoplasm. We can have benign neoplasms, an abnormal growth of cells, just too many replications, too many times going through the cell cycle. I'm getting lots and lots of cells. Benign neoplasm or tumor is usually encapsulated, and those out of control replicating cells don't usually invade or spread to neighbor tissues. So neoplasm is a tumor. It's uncontrolled cell division. So mitosis has just gone wild. So those checkpoints and those signals that we were getting from neighboring cells saying time to divide or time to stop dividing, we're just getting the divide, divide, divide information or signals. Malignant neoplasms are cancerous. They are not encapsulated. So here I have just these out of control dividing cells. There is no capsule. There's nothing separating them from the surrounding tissue. So they readily invade and get into those tissues. These balls of dividing cells, these masses of dividing cells, usually have a ragged edge as opposed to here, since they're encapsulated when they're benign, it's a smooth edge. And the outer cells easily break off or detach from this mass and move, and can move to distant places, settle, and start undergoing uncontrolled cell replication. That's metastasis. When a cancer cell metastasizes, it has moved to elsewhere in the body and begun undergoing unregulated cell division. This can result from a mutation in the genes that regulate the cell cycle. So those proteins, those signals that are coming, that are telling the cells to divide or stop dividing, those are regulated by genes. So on our DNA, we have genes that code for proteins. Some of those are those signals. And we have genes that say, okay, we should move forward now building this protein or that protein, or we shouldn't. So we have some sort of message in the regulation of the cell cycle that is faulty. So there's something that either said, stop dividing, or we have enough cells that has gotten turned off, and so we keep thinking we need more cells, or a regulator that says, hey, keep dividing until I tell you to stop, that never says stop. Carcinogenesis is the development of cancer. It tends to be gradual and takes many, many years before that error or damage in the cell is noticeable and obviously cancerous. So our characteristics of cancer cells is they lack differentiation. Typically our cells, every cell in our body has all the DNA with all the information for everything our cells should do. But at some point, they know what they're supposed to do. I don't need all the information. So I don't need all the recipes for everything my cell could possibly do, only for the things that my body could possibly do, only for the things that I, as this type of cell, do. And so they turn off a number of genes. They differentiate. So Every one of my cells is a stem cell, has the potential to be a nerve cell, but at some point it differentiates and says, I'm a nerve cell, I'm going to turn off all these other functions. I don't need to have actin for movement, for muscle contraction, because I'm not a muscle cell. I don't need to do these other functions. I'm not going to secrete anything, and this is what I'm going to do. And so we say it's differentiated. It knows what it's going to be when it grows up, and it's just going to use those genes. Cancer cells never get that message. They never differentiate. All of the DNA is active. They have the potential to be anything, but all they do, they don't really go to that G1 phase and do a job. They just keep multiplying. They tend to have very abnormal nuclei. They might have lots of nuclei in one cell, or misshapen nuclei. They form tumors, which are masses of uncontrolled replicating cells. Typically, mitosis can also be controlled by contact with neighboring cells. This is called contact inhibition. I have the right density of cells. We're touching at the right pressure. We're done. We're not going to divide anymore. Cancer cells don't have contact inhibition, so it doesn't matter how tightly they're pressed up against their neighbors. They're going to keep making copies. They can undergo metastasis, fragment off, and spread. And cancer cells can also undergo angiogenesis. So blood cells, typically with benign tumors, there is no blood cell, there's no vascularization, there's no blood cells feeding them nutrients, oxygen, taking away wastes. But with cancer cells, their blood vessels do develop. We get new blood vessels growing through there, so they continue to nourish those cells that otherwise, if they were just a mass with no vessels feeding them, would starve. So we have our comparison. Normal cells are differentiated into what they're supposed to do. They know their job, and that's all they are going to do. Cancer cells are non-differentiated. We have normal nuclei, pretty much like all the pictures we've seen. We have abnormal nuclei. They can be misshapen. They can be too large, small. We can have multiple in the same cell. These never get that signal for apoptosis, to commit suicide. They don't know there's something wrong with them. Normal cells, if there's an error at any of those stages in the cell cycle, they get a notice to undergo apoptosis. Here we have no contact inhibition, just continued growth. Here, once we have the right number of cells that we need in a certain area, we stop. And these are nice organized layers. I could take a cross section and see this lovely organization like stacking bricks. Whereas cancer cells are disorganized, multi-layered, just masses. And cancer cells will undergo metastasis and angiogenesis. So this is normal cells, sort of this regular, spaced out, same size. And cancer cells that can have all different sizes, some with multiple nuclei, some with larger, smaller. I've got 2 nuclei there. So very, very specific or different in appearance from what normal cells look like. So cancer, where does it come from? Well, some cancers are genetic. We have oncogenes. If we have a mutation in our DNA repair mechanisms, so we have these processes that can repair. If we have an error in our DNA replication, we can correct those errors. We can get a mutation in the part of the DNA that codes for that. Oncogenes are cancer-causing genes. So proto-oncogenes, these are genes that promote the cell cycle. Tumor suppressor genes inhibit the cell cycle. So normally, we have ways to keep the cell cycle going, to suppress out of control replication, and both normally are regulated in coordination with the organism's growth plan. So hormones and chemical signaling say, yes, time to grow, no time to stop. If these mutate, either one of these, we can either get uncontrolled growth or some other error in that cell cycle. Telomerases, chromosomes usually have special material at each end called telomeres. Telomeres are the end parts or the far parts. And every time the cell divides, these get a little shorter. When they get very, very short, the cell no longer divides. So it's like we have so many tickets in our cell for divisions. When you run out of tickets, you don't get to divide anymore. Telomerase is an enzyme that adds telomeres to that line. So it's like buying new tickets. If that enzyme is mutated, if the gene that codes for that enzyme is mutated, then we keep getting new telomeres and our cell keeps getting tickets to divide. So like counterfeit tickets. The cell keeps getting these tickets. I can divide, I can divide, I can divide, when they should normally stop at some point. And that covers everything in this chapter. So, and that covers our unit. So we are through unit 3, and our exam will cover chapters 6, 7, 8, and 9, and that's on Thursday. It'll be available between Thursday and Saturday. I will post another video with a review on Thursday for you. So if you're ready to go before then, the exam opens at midnight Thursday. So you can take it before then, but I should have that posted by mid-morning for you. All right, thank you.